Hi, this is AQU239 Larvae Food Production. We will presenting about effect of different media in the growth of algae. My name is Azril Mikael bin Azim. This is my team members. Next, this is our table of content.
Hi, my name is Sofia Cristina. I will explain about microalgae. As we know, microalgae are the most prevalent primarily unicellular producers, and they can be found in freshwater, saltwater, hypersaline lakes, deserts, and even Arctic environments. In aquaculture, microalgae are an important resource because they provide fish larvae, shellfish, and crustaceans with high quality feed. The nutritional profile of aqua feed is improved by microalgae, which are abundant in proteins, fats, and important fatty acids like omega-3.
Next, I will introduce you about NPK fertilizer. Fertilizer labels display NPK values because nitrogen, phosphorus, and potassium are the key macronutrient essential for plant growth. Each serve a specific purpose. Nitrogens promote leafy growth, phosphorus support roots and flowering, and potassium enhance overall health and resilience. Understanding NPK ratios help you to choose the right fertilizer to meet your crop needs.
Introduction: Fish meal. Fish meal is a powerful nutrient source for algae culture, promoting growth because of its high protein and vital nutrient content. In aquaculture, fish meal is often added to the algae tank to promote a faster biomass production.
So, for algae culture, we will start with preparation of algae starter. The preparation, we will start with providing an aquarium with structure and label T1 till T3. Next, put water half full inside the aquarium and add the algae solution from the fish tank to the aquarium for algae culture. Lastly, provide aeration and light source for the algae culture.
Preparation of fish meal. The first step: put the trash fish in the drying oven for 1 week. Fish need to turn upside down every day. This step to ensure that the water moisture in the fish are decreased. The second step: grind the dried fish into a fine powder. The fish meal was put in the drying oven for the second time. The reason why the drying process needs to be done for the second time, it is to make sure that there are no water left in the fish meal. This process will ensure that the life of the fish meal will last more longer.
Method: counting the algae cell. Firstly, the algae sample was taken from the hatchery and then one representative will send the algae sample to laboratory. Next, the algae sample was mixed with legal solution before we observe the algae under microscope. A Neubauer hemosimeter was used for counting microalgae cells. The hemosimeter slide need to be cleaned first before putting the mixed algae suspension on it. For the next step, the presence of air bubbles overflowing, underfilling and uneven distribution of cells need to be avoided by put 1 drop of well-mixed algae suspension on each Neubauer chamber and cover with its cover slip. Then, the mixed algae suspension will be observed under the compound microscope. After that, the picture was taken as a result. Lastly, the cell density will be calculated using the respective formula.
Next, I will explain how we putting the NPK fertilizer as media in tank 2. First step is the NPK fertilizer was weighed according to the measurement standard of 0.2 mg, which was also used in past research. In past research, 1 liter was measured as 0.01 mg. Second is the NPK fertilizer was mixed with water from tank 2 and crushed until well blended. Lastly, For tank 2, insert 0.2 mg of NPK fertilizer into each replicate that we have done in step 1. Next, for the tank 3, we put fish meal as a media. The methods are the same with the NPK fertilizer. So, the first step is the fish meal need to be weighed according to the measurement standard of 0.2 mg. Next, the fish meal was mixed with the water from tank 3 and crushed until well blended. Lastly, for the tank 3, insert 0.2 mg of fish meal into each replicate that we have done in first step.
Result on initial algae cell. We did not take the initial sampling from tanks T1, T2 and T3 because the algae that we capture in the bottle for 1 week using algae starter pack died when we observed it using hemocytometer. Then, we used the algae straight from the fish tank rather than properly culture them. So, we don't have the precise data to demonstrate how much the algae were in the tanks at the beginning without a proper initial sampling.
Result on number of algae cell week 1 to 3. Week 1, the average number of algae cells for treatment 1 is 18.33, treatment 2 is 21.83, and treatment 3 is 14.52. Next, for week 2, the average number of algae cells for treatment 1 is 39.86, treatment 2 is 29.68, and treatment 3 is 27.47. Lastly, for week 3, the average number of algae cells for treatment 1 is 13.05, treatment 2 is 16.08, and treatment 3 is 34.32. Result on final number of algae cells. The average final number of algae cells for treatment 1 is 28.4, treatment 2 is 48.8, and treatment 3 is 69.0. Based on the graph, the best performance is treatment 3, which is we are using fish meal. Fish meal treatment can be beneficial for algae growth due to several reasons. First, it is rich in essential nutrients such as protein, amino acid, and fatty acid. Other than that, fish meal also high in protein, which provide a readily available source of nitrogen. Source of nitrogen is a key nutrient for algae growth. Lastly, fish meal has a balanced nutrition such as contain a balanced mix of macro and micronutrients including vitamins and minerals which support various metabolic processes in algae. Algae growth dynamic graph. For this dynamic graph, we didn't get a dynamic graph like in the notes given, which have 5 growth phases of microalgae cultures. For treatment 1, algae growth dynamic graph is going from induction phase, then going to exponential phase, and lastly going to death phase. While for treatment 2, algae growth dynamic graph is going from induction phase, then going to exponential phase, then going to death phase, but lastly, it's going back to exponential phase. For treatment 3, algae growth dynamic graph is the same as treatment 1, which is it's going from induction phase, then going to exponential phase, and lastly going to death phase.
Finally, the conclusion. To conclude, this experiment demonstrates the effect of different growth media such as NPK fertilizer and fish meal on algae development. It highlighted the importance of proper preparation, media composition, and monitoring for optimizing algae growth. The finding contribute to sustainable practices in aquaculture by using accessible and cost-effective resources. By the way, this is our challenges. Now, let's talk about the challenges we faced during this experiment. First, The lake water from UiTM Muka did not contribute positively to the algae growth. The water might have lacked the necessary nutrient or contain elements that inhibit growth. Next, we face an issue with insects frequently getting into the aquarium. This disrupts the experimental environment and could have affected the algae culture. The third one, there was a lack of consistent aeration towards the end of the experiment. Proper aeration is essential for maintaining a healthy environment for algae growth, and this limitation may have impacted our result. Moreover, there was no 24-hour surveillance of the setup. This meant we could not address problems such as equipment malfunction or environmental changes in real time. Lastly, we faced difficulties in accurately counting the algae cells. Issues like air bubbles, uneven cells distribution on the hemocytometer, and inconsistent preparation of sample affected our ability to obtain precise measurement, and the microscope we used was not very clear, which made it difficult to observe and count the algae accurately. This impacted the precision of our result and added to the challenges of data collection. These challenges provided valuable lessons and highlighted area for improvement in future studies, particularly in refining our method and equipment.
So, for the future direction of the study, first is utilizing widely accessible and reasonably priced materials as algae growth media to lower manufacturing costs without sacrificing quality or growth rates. Second is examine the best ways to preserve the nutritional content of algae that collected from various media, such as drying or freezing them. Lastly, cultivation by growing algae together with beneficial microorganisms in the same environment to see if it increases algae growth or nutrient content to ensure a natural ecosystem.
Implication of the study. This experiment taught us how various treatments such as fish meal and NPK fertilizer can impact algae development. From starting with a little sample to monitor its growth using instruments like hemocytometer, we learned how to culture algae step by step. This experiment really helped we get better at what we can do. We can now precisely monitor growth, make algae culture, and how to count algae cells in the process. We also improve our group collaboration and problem-solving skills.
In conclusion, we can see that we can recognize how important media composition is for maximizing algae growth and its possible use in aquaculture. Next, a deeper understanding of creative methods and sustainable practices in the manufacture of larvae feed. Lastly, we can acknowledge the importance of flexibility and trial and error in research to successfully handle unforeseen obstacles.